We will now further discuss functions and activities of vitamin D, focusing in this portion of the presentation primarily in the role of vitamin D in calcium homeostasis. We also discuss other functions of vitamin D as well as the RDA and UL levels. So one of the primary functions of vitamin D is in its ability to regulate serum calcium levels. We talked about with serum calcium that it's very important to maintain serum calcium levels within a tight range between 8.5 to about 10 milligrams per deciliter. We see that vitamin D then plays a role in calcium homeostasis at the level of the intestine, the level of the kidney, and the level of the bone. So we'll be talking about the specific mechanisms of how vitamin D is going to play a role in calcium homeostasis at these tissue sites. We know that vitamin D is going to work with parathyroid hormone and respond particularly when serum calcium levels are low. And so the effect of parathyroid hormone and active vitamin D activity is to increase serum calcium levels. So we see overall at the level of the intestine, active vitamin D or calcitriol will increase calcium absorption. We'll see that active vitamin D or calcitriol at the level of the kidney is going to increase calcium reabsorption and decrease calcium excretion. And then at the level of the bone, calcitriol is going to increase the demineralization of bone and thus freeing calcium from the bone so that it can enter the bloodstream. So each of these effects are going to promote an increase in serum calcium levels when serum calcium is low. So we'll look more specifically at the mechanism at each of the body sites, so at the level of the intestine, the kidney, and the bone, to see how more specifically vitamin D has an effect. So we talked about vitamin D's binding to its nuclear receptor, and in response to the binding to its nuclear receptor having effect on DNA activity, specifically on transcription and protein synthesis. So it's going to be this effect and its binding to the nuclear receptor, which is going to increase the synthesis of key proteins involved with the process of absorption of calcium at the intestine, reabsorption of calcium at the kidney, and also the proteins involved in the demineralization of bone. So now here at the level of the intestine, we're going to talk about the effect of calcitriol. So in the intestine, we have the nucleus of the intestine, and within the nucleus of the intestine, we have the vitamin D receptor. So we see the vitamin D receptor found in the nucleus of the intestine, and then calcitriol will then enter the nucleus and then bind to the nuclear receptor within the enterocyte. So in response, we see generally an increase in transcription and increase in protein synthesis, and the proteins that will be synthesized in response to calcitriol binding to vitamin D receptor are going to be those proteins involved in calcium absorption. So we learned previously that we have this process of calcium absorption. We have the TRPV6 channels used to bring calcium through the brush border membrane. We have a protein called calbindin that shuttles calcium from the brush border to the basolateral membrane, and then we have another protein called the calcium ATPase. So each of these proteins, the TRPV6, the calbindin, and the calcium ATPase involved in calcium absorption, the synthesis of these are upregulated or increased when the vitamin D in the form of calcitriol binds to its nuclear receptors. So thus, if we have more channels, if we have more of this calcium binding protein, if we have more of the calcium ATPase, that increases our ability to absorb calcium. We see in this diagram more detail on the nucleus of the enterocyte. We see the DNA strand and then we see here with the vitamin D receptor calcitriol complex, then that's going to promote an increase in transcription, increase in protein synthesis, allowing for enhanced ability to absorb calcium. It will be important to note the orientation of this enterocyte. So the orientation here is different than the orientation here. So in this diagram that we've been going over previously, the brush border membrane is here, where we have the lumen of the small intestine and then the brush border and the absorption. But we can see here that it's flipped. So we have the brush border membrane here, the lumen of the intestine we would say would be here, with calcium entering the enterocyte and then going through the enterocyte bound to calbindin and then out through the basolateral membrane at this portion. So just to keep in mind that the cell here is flipped, and this is just to convey how when we have the circulating vitamin D, the calcitriol in the blood, then we see that it actually enters through the basolateral membrane, and then it binds to the vitamin D receptor, and this is going to be in the nucleus of the enterocyte, and then the additional processes leading to the increased transcription, increase in protein synthesis. And this protein here is specifically going to be that calbindin that is used to shuttle calcium through the cell. So next we'll talk about the activity of calcitriol at the level of the kidney. So we said previously that we have calcitriol promoting calcium reabsorption. So it increases the reabsorption and then decreases the excretion of calcium from the intestine. So through urine, it decreases urinary calcium excretion and increases the reabsorption of calcium. So we'll see that there's also the same mechanism with calcitriol binding to nuclear receptors in the kidney that's increasing gene expression and protein synthesis of calbindin and the TRPV5 channel. So we have similar proteins in the kidney that we saw present in the enterocyte. So this TRPV5 channel, so this you can note is different than the TRPV6 found in the enterocyte, but it's a similar structure. We have the TRPV5 and calbindin promoting this reabsorption of calcium at the level of the kidney. Also, we will see the effect of calcitriol at the level of the bone. So at the level of the bone, we will also see the binding of calcitriol to nuclear receptors. However, the mechanism is a little bit more complex, involving compounds called rank ligand, R-A-N-K, or rank ligand, and also what's called a rank receptor. So the synthesis of these compounds are going to be involved in regulating the demineralization of bone. So here describes the process, and then we have an image that we're going to review. But we'll see that at the level of the bone, we have both osteoblast cells and osteoclast cells. Osteoblast cells are involved in building bone or forming bone, and osteoclasts are involved in demineralizing bone. So we've said that when serum calcium levels are low, calcitriol and also parathyroid hormone together are going to act to increase the demineralization of bone, which will allow the mobilization of calcium into the blood. So we're going to see that the overall effect of the calcitriol is to increase osteoclast activity, which is going to promote this demineralization. So we'll talk about the mechanism of how this occurs. First, we'll see that calcitriol will bind to nuclear receptors first on the osteoblast cell. So it will bind to nuclear receptors within the osteoblast cell, and the osteoblast cell is involved in increasing the synthesis of what's called this rank ligand. So the rank ligand that is formed from the osteoblast cell binds to rank receptor. So we have this ligand receptor binding. The rank receptor is present on the outer portion of the osteoclast cell. So when rank ligand binds to rank receptor on the osteoclast that will promote the activity of osteoclast. So it's somewhat of an indirect effect and function of calcitriol on the osteoclast because it actually interacts with the osteoblast cell with the binding to its nuclear receptor, increasing the rank ligand, which then will bind to the rank receptor on the osteoclast, and this binding of the ligand to the receptor will increase osteoclast activity and the demineralization of bone. So we see here a diagram of this process. And so we will see that with different factors, so one of them being this calcitriol, so we have calcitriol binding to nuclear receptor of the osteoblast cell, and that in turn is going to increase the synthesis of this rank ligand. So this green structure is the rank ligand, so the calcitriol binding to the nuclear receptor of osteoblast will then form the rank ligand, and then we see in blue here this rank, and the rank is going to be existing on the outer portion of the osteoclast cell. So when the rank in green, the rank ligand in green binds to the rank receptor, then that in turn is going to activate the osteoclast. In this diagram, we see an integrative picture of the effect of calcitriol and parathyroid hormone at the level of the intestine, the kidney, and the bone when serum calcium levels are low. So this just reviews each of the effects that we've discussed in terms of the effect of calcitriol at each of these tissue sites to increase serum calcium levels. So you can take a few moments to review this. You see that they have emphasized the activity of both PTH and calcitriol, but for the purposes of this class, we're going to be focusing primarily on the effect of calcitriol at each tissue site, but PTH is going to be very key and integral in converting vitamin D to that active form. So with PTH, it promotes conversion to calcitriol, and then we see from there calcitriol is going to have its effect at the various tissue sites, increasing blood calcium levels. We see additional effects of vitamin D. We see that in addition to having an effect on regulating calcium homeostasis, that there's an effect on cell differentiation. We see that it limits proliferation of different cell types and also limiting growth of different cell types. We see an effect on blood pressure. So we will discuss further when we talk about sodium and potassium, what's called the renin-angiotensin-aldosterone system. But just generally, we see here that it reduces renin and angiotensin, thus reducing blood pressure. Also, we see that there are beneficial effects to reducing the incidence of different autoimmune disorders. It promotes more normal insulin secretion and also promotes normal muscle function by binding to nuclear receptors that are involved in upregulating calcium ATPase pumps, which promotes movement of calcium into the sarcoplasmic reticulum. So you won't need to know all of the details of this, but just generally be familiar with the fact that vitamin D is also involved in muscle function based on its effect on the calcium movement into the sarcoplasmic reticulum. We see here that RDA for vitamin D is 600 international units per day, and this actually is a more recent RDA that was changed in 2010. So prior to 2010, vitamin D was an AI level, and it was 200 international units per day. It was the AI for vitamin D. So the RDA has been increased as compared to the AI in adults, and then in those over 70, it's even higher at 800 international units per day. We see the tolerable upper intake level being 4,000 international units per day.